A black background with white writing that says Georgia and Kat's accessible audio-described science videos with an exclamation mark at the end of the sentence. The words Biology Core Practical 1 onion cells are under it. There is also a picture of a microscope.
Hello and welcome to our accessible audio-described science videos. I'm Kat.
And I'm Georgia.
Today we are going to be describing what it is like to look at an onion cell under a light microscope. This is one of the core practicals for GCSE biology.
There is a picture that is on the screen now of a light microscope. The microscope is standing on a short, straight metal cube, which is called the foot or the base. Above this, there is a square, which comes out of the arm of the microscope, which is called the stage. The stage has got metal clips on it to hold the slide in the right place. The slide is a clear glass rectangle, which is placed over a hole in the stage, where a light comes through. The arm is a curved semicircle, which feels like the handle that is on a sliding door. It is on the side of the microscope. Attached to the arm, there is a focusing wheel, which feels like the wheels on a toy car. The arm is also connecting to a hollow cylinder, which has the eyepiece lens on the top of it. The eyepiece lens is directly lined up to the hole that is in the stage. At the other end of the cylinder, there are 3 objective lenses, which rotate and increase in size from the smallest magnification to the largest magnification.
The first stage of this practical is to prepare the onion. The onion is an example of a plant cell.
We are now peeling the skin that is from the layers of a piece of onion. The skin is very thin and almost transparent. It feels like the skin of your earlobe.
This layer of onion skin is exactly 1 cell thick, which is why we can use it. It allows the light to come through. We are now going to place it on the slide.
We are using forceps to place the onion skin onto the slide. We are then using our fingers to flatten the onion skin. In this video, there is a fold that is in the middle of the onion skin that we can't use when we are looking at the cells with the microscope, but we have enough flattened skin on both sides so it won't affect the experiment.
Next, we are placing a drop of iodine stain on the top of the onion, which will show contrast between the different parts of the cell.
We are placing an orange and brown solution on top of the onion skin, which is now on the slide. We are using a pipette to do this, and we have squeezed it too much, and so some of the iodine has gone onto the table, but there is enough iodine that has gone onto the onion skin.
Next, we will cover the slide and the onion skin with a cover slip to keep the skin in place.
We are now lowering the cover slip, which is a very thin, small square piece of glass, from one side of the onion skin to the other side of it, using forceps, which are sciencey tweezers. The slide is on a piece of white tissue, and we are folding it over and gently pressing it down. The excess iodine has made the tissue go black because the tissue contains starch, but we will talk about why it happens in our video that is going to be about food testing.
Now we have prepared the slide and we are going to put it under the microscope.
We are now gently placing the slide with the onion skin on it on the top of the stage and placing the stage clips onto the slide to keep it in place.
The eyepiece lens has a fixed magnification of ×10, but the objective lenses are varied, so we are going to pick the lowest magnification to give us a wide view of the onion skin.
We are turning the rotating circle which holds the objective lenses. The smallest objective lens is now directly below the eyepiece lens.
Now we will use the focusing wheel to focus the image which is in the eyepiece lens. We start with the objective lens as close to the slide as possible, then put our eyes over the eyepiece lens and focus the image by turning the wheel away from the slide.
We are turning the focusing wheel with our eye over the eyepiece lens. The image is becoming gradually clearer to reveal lots of onion cells. They are all regular rectangle shapes. The iodine has stained the cell wall and the nucleus, showing a black dot in the middle of the cell.
Now we have found the cells, we are going to look closer at them by using a bigger objective lens.
We are turning the objective lenses again so that the medium-sized lens is below the eyepiece lens and making small adjustments to the focusing wheel. And now we can see the same cells, but they are larger.
Now we're going to pick one of the cells to draw a scientific drawing with. It's important to pick a sharp pencil and to draw exactly what you can see, but we're going to draw it on an iPad, which is a more accessible way.
A white rectangle with circular corners is being drawn on a black background, and then a small circle is drawn in the centre of the rectangle. Then 3 lines that are horizontal are drawn on the outside of the rectangle using a white ruler, and they are labelled with white writing. The first line that is pointing to the sides of the rectangle is the cell wall. The second line that is pointing at the small circle that is inside the rectangle is the nucleus. And the third line that is pointing to the black part of the inside of the rectangle is the cytoplasm. There is now a picture of the onion cells with the largest magnification that is on the screen. There are some examples of some exam questions that are about this practical that are in the description of this video, which is under the title that is for the video.
Write your answers in the comments and watch our video next week to find out if you got the answers right.
We hope that you have found this video accessible and interesting.
Please feel free to leave us a comment. And don't forget to subscribe for more accessible content.